Hi, so today we're going to talk about a little something called protein synthesis, okay? And there's three parts to this that we're going to be talking about today. There's transcription, which is what you're looking at right now, translation, and there's also mutation. Now mutation's a little more in-depth and it's a little different, so we're going to go through transcription and translation first. So transcription actually occurs in the cytoplasm instead of the nucleus, and there are different things involving. There's your DNA, which provides the genetic information, and it basically decides how things are going to happen. There's also the genetic code, which is this kind of thing right here, and that's the information for synthesizing. So I know you guys do know a little bit about cellular reproduction, and this is a little bit different and a little bit more in depth. So what we're going to be focusing on with transcription is messenger RNA, which carries the gene messages out of the nucleus. RNA is a little different than DNA. It's single-stranded instead of double-stranded, and it's a lot shorter than DNA. Okay, so the genetic information reaches the cytoplasm by being copied into RNA from DNA, okay? The process of synthesizing is transcription, and they have to follow the rules of complementary base pairs. So as you see, G goes to C and T goes to A. It can go GC or CG, and it can go TA or AT, but it has to follow the complementary base pairing. So C can't go to T and A can't go to G. It has to be G to C and T to A. Okay, so now that we understand transcription a little bit better, we're going to go a little more into translation. So translation is a series of codons on mRNA transferred from one language to another language. So these different languages are nucleic acid and amino acid. So they're two different basically sequences. The sequence AUG represents the start of a gene, so you can kind of see that represented here. The mRNA molecules leave the nucleus and associate with a ribosome in the cytoplasm. Building a protein molecule requires a connected amino acid in the cytoplasm, positioning them in order specified along a strand of mRNA. A second kind of RNA molecule called transfer RNA. And as you can see here, transfer RNA synthesized in a nucleus and sent to the cytoplasm, and it's assisted in the protein construction. Correctly aligned amino acids are linked by enzymes to form proteins. tRNA, so transfer RNA, is synthesized in the nucleus and sent into the cytoplasm. It assists in the construction of the protein molecules, as we said before. There are 20 different types of amino acids from the biological proteins. So there have to be 20 different types of tRNA molecules, and they must be available. Each tRNA has a region at the end that is 3 nucleoids. They form complementary base pairs with the RNA codon, which are called anticodons. And a codon is a set of 3 nucleoids of mRNA that specify an amino acid. This is how they are correctly positioned. And it occurs in the ribosome, as we said before. As the process begins, ribosomes bind on the mRNA molecule. A tRNA molecule, so once again a transfer RNA molecule, with complementary anticodons bind. A tRNA with complementary anticodon holding its amino acid form hydrogen bonds with the first mRNA codon, so that first strand that we saw. A second tRNA bind to the next codon, bringing amino acids to an adjacent side on the ribosome. A bond forms between the 2 amino acids, beginning a chain. The first tRNA is replaced from the amino acid. It's regulated to the cytoplasm. This process repeats. Enzymes control the protein synthesis. As the protein forms, it holds on to the unique conformation and is released. Correctly folding it is essential. A gene that is transcripted and translated into a protein is said to be expressed. Okay, so the last and final thing we're going to talk about today is mutation. So codons are read in sets of 3. So ATC, ATC, TTT, GGT, TCC. Everyone has to be read in 3 sets. So most often what you're going to see is a missing letter or an added letter, and when that happens, that can change the entire sentence, basically. You can almost think of it as if it is a sentence, and if an extra letter was added, the words would no longer make sense because every letter would be shifted down by 1 because it still has to be read in that 3. So if you were to add an extra letter right there, that would now be ATC, ATC, ATT, TGG. It would completely change the sentence. Or if you were to delete a letter, then ATC, ATC would become ATC, ATT, TDG, GTT instead of what you see there. When you do insert or delete these bases, it can actually change the entire sentence and the entire codon sentence. Sometimes when the last letter is changed out for its counterpart, so a G is changed for a C, if it's at the end, it can actually have no effect, and it's called a silent mutation. However, you could change you could get a code that changes it to a stop codon, which can cause the strand to actually be too short, in which case it probably won't function properly if it functions at all. So there are a few different types of diseases that are actually caused by mutations. There are many, but a few are cystic fibrosis. There's also anemia as well as hemophilia. But what you're looking at on the board right now is cystic fibrosis. So as you can see, there's a deleted base, so this C does get deleted, which causes ATC, ATC, TTT, GGT, TTC, which is the ideal codon, to turn into ATC, ATT, GGT, TCC. Now, when you're looking at that, that doesn't seem like it's a big deal. You're saying it's only one letter, that's all that's missing. But if you know what cystic fibrosis, which is actually when you basically feel like you're drowning because there's too much water or thick mucus in your lungs and you can't breathe, you wouldn't be saying that missing gene is just a small thing. It's actually amazing how one small change in a genetic code can cause such a big difference in everything else that surrounds it. And that is protein synthesis.